Good morning, good afternoon, good evening. Welcome everybody to a new Global IMMUNOTALK. I am Carla Roslin, and I would like to remind you that next week we'll have Marion Pepper with us, so join us also next week. And I'd also like to share with you that we have started to go global live. So for the talks of our global immuno speakers from Australia, they will go live on the Wednesday that they are scheduled at 4 p.m. Australia time, but we will be connecting again like every week at the regular time on this other side of the world. And this is quite similar to what happened yesterday. So our speaker of today, Shalin Naik, connected with Elena yesterday for us, and I will now try to share with you that talk. I think you will also appreciate some nice things that happened to Shalin and his wife.
Hi everyone. I'm delighted to be introducing to you today Dr. Shalin Naik. Training with Professor Tom Schumacher. And after that, he came back to Australia, to the Walter Eliza Hall Institute, where he became a group leader and he started his independent laboratory. Shalin has received many awards for his amazing scientific track record. This includes, but of course is not limited to, the Premier Commendation for Medical Research Award from the Victorian government in Australia, the Young Investigator Award from the DC Terra Centers of Excellence, and many other ones that we don't have time to go through. So Shalin's laboratory focuses on the study of how the cells of the immune systems are formed from blood stem cells. His team utilizes new technology that interrogate the individual cells rather than the cell population as a whole. For that, his laboratory takes a single cell systems biology approach that have enhanced our understanding of how hematopoietic stem cells ultimately generate all the red and white cells of the immune system. Indeed, by using cutting-edge technologies to study single cell at different functional and molecular levels and integrating this information, Shalin's team has revealed important mechanisms behind hematopoietic sulfate specification in health and disease. And many of these findings have the potential to unlock strategies for manipulating blood stem cells and may have future applications for stem cell therapy or immunotherapy and provide insights into cancer formation. So I'm sure we will learn much more about Shalin's scientific program today during his Global IMMUNOTALK that is entitled Clonal Multi-omics in the Study of Hematopoiesis. So Shalin, thank you so much for accepting to be a global immuno speaker, and thank you even more for making it work. I know that you will introduce now why your life is going through a very special times today. So thank you so much for making it work. So as always, Shalin, before I ask you to share the slides, I would like to ask you a question. And the question we want to ask you today is related to your role as TV presenter in the Ask the Doctor and the Catalyst series that are in ABC TV and Netflix. And so this is more atypical for a scientist to be a TV presenter and host. And so the question is with how these roles as TV leaders and hosts have impacted or contributed to your scientific career.
Thanks, Elena, and the organisers for inviting me to talk here. It's really a prestigious seminar series, and I feel very fortunate to be amongst the names that I've seen come across here. Yeah, the TV thing kind of came by accident, really. I've always been interested in science communication, in addition to the science research itself. And this call came along to our institute. They said, if anyone's interested in hosting a show, you can send a little 1-minute clip. So I recorded myself on my phone talking about stem cells, and they liked it, and I did an audition, and then I became a host of a TV show. And for all non-Australians, you can catch it on Netflix. It's called Ask the Doctor. So you can check it out. It's a fun lifestyle, health and science program. And did it help my career? It took me out of the lab quite a lot. So in that way, it didn't help it. But I think communication is really 50% of science, in my opinion. 50% is doing the good science, and 50% is being able to communicate that idea to not only other scientists, but the general public. So I believe it's really important. And yeah, and well, you'll be able to judge me on my talk whether that's indeed true.
I'm sure it will be a wonderful talk, Shalin, and for sure we will check out the show in Netflix. And so definitely looking forward to that. So now we are all ready. So if you'd like to share your screen. Let's see.
There we go. How's that work?
Perfect. Fantastic.
Wonderful. All right, well, look, thanks once again for the invitation to speak. And the special circumstances under which I'm speaking are that I'm in the maternity ward at a hospital in Melbourne because just this morning our second son was born, and I was going to cancel, but my wife, who's also an immunologist, said, no, you have to give this talk. Go, pre-record it in case, you know, we can do it at a time that's convenient. So apologies for the pre-recording, but I hope you can forgive me. So the subject that I wanted to talk about today is really culminated in one paper that's just come out in Immunity. And that's around clonal multi-omics in the study of hematopoiesis. And this graphic is actually from a barcoding paper we had in cancer, where each dot is a clone that contributed to the development of cancer, and each column is a different section of tissue. But the principles are the same in that I've always been intrigued into as to how a single cell determines its fate clonally. That is, how does one cell make two or more cells of anything? It could be a naive T cell making an immune response. It could be a stem cell making an entire tissue. Or it could be one of the most important single cells of all of us today, the original zygote, which has in it, and just think about that for a second, the program for the entire human. I think sometimes we forget that, how elegant and programmatic these cells are. And so how can we decode what a single cell can make in using novel molecular tools, and how can we use that to understand biology and disease? And that's really the subject of my lab. So we all start from a single cell, and then that cell divides 2 and then 4 and 8, 16, and ends up turning into a series of tissues. And if those decisions, if those division events did not make a decision to go one way or the other way, we just turn into amorphous blobs. But we're not amorphous blobs. We are structured humans with eyes and ears and mouth and nose and a heart on this side and a liver on that side. So that patterning must occur somehow, stochastic or deterministic. That's up for debate for many things. And then you get something like this. And this is our new arrival. He just came this morning. We don't have a name for him yet. But he's just saying, hi, Global IMMUNOTALKs. So how do cells make decisions to make one thing or another thing? And really, at the end of this tree is not just a bunch of cells. They are highly specific tissues with highly specific cell types within those. And in fact, as we know from all of the single cell papers that have come in the last 10 years, if you take a piece of tissue and you just study it in bulk RNA sequence, bulk proteomics, you're looking at the smoothie of that tissue and not the individual fruits. And unless you look at the individual fruits and nuts, you don't know whether a particular cell type even existed or not in a particular tissue. And the ionocyte in the lung is one of the classic examples, the recent examples of that. So single cell technologies have been around since we knew about cells, really, since we first looked at cork cells under the microscope. We've been doing single cell biology, let's be honest. But really, it's these new techniques of single cell lineage tracing and single cell RNA-seq that I'm interested in, and not just cataloging cells and finding differential expression patterns, but taking that information and trying to get some validation of novel biology out of that. Instead of, cataloging is really important, really useful, like efforts in the Human Cell Atlas, but obviously we want to take it the next step and find something from that. But let's not talk about single cell RNA-seq, let's talk about clonal heterogeneity. So if you had a population of 10 stem cells, and those 10 stem cells made red, green, and blue cells, you could conceive of actually an infinite number of ways those cells could be made from those 10 cells. I'll give you 3 examples. Those 10 cells could each make red, green, and blue cells equally. Alternatively, a subset makes red, a subset blue, and a subset green, or there's some sort of heterogeneity in their output. And really trying to answer this question has been really the trajectory of my career for the last 15 years. So in classic models of hematopoiesis, which I'm sure many of you have seen, you have all of the immune cell lineages, hematopoietic cell lineages on the right, and they are assumed to come from these kind of ball and stick diagrams where one population turns into another. And the underlying assumption is that every cell within that population is equal, all right? We know in reality it's probably not equal, but the assumption is that it's equal. And so if we just zoom into the early stem and progenitor cells here, and let's just focus on the multipotent progenitor, otherwise known as MPPs, and in today's talk, the MPP4s. The assumption is every cell in that population is capable of making clonally lymphoid and myeloid output, or lymphoid and myeloid and erythroid, the spectrum of lineages. But is that really the case? So, I set about this question at the end of my PhD in 2007 to really ask, how can we assess the output of hundreds of single cells in vivo? And so, of course, you could inject one cell into one mouse, and many people have done heroic studies doing just this. But what if instead you could barcode those individual cells and inject them into one mouse? Good for the mouse, good for the researcher. So we, in fact, did that in a paper during my postdoc with Ton Schumacher, and this is quite a while ago now. We took barcoded lentivirus, hematopoietic progenitors, we transduced them, injected them in vivo, and now the barcode in the cell is inherited by the divided progeny, such that at the end of the experiment, you can sort cells from cell type A and those from cell type B, lyse them, and do a barcode PCR and sequence those barcodes. And now if you find barcode 1, for example, let me get my red cursor now. Yeah. Sorry, laser pointer, here we go. If you had barcode 1 present only in cell A but not cell B, you would assume there was a mono outcome progenitor here that gave rise to only cell type A. If you find barcode 2 reads in cell type A and B, it means it came from a common ancestor, and so on and so forth. And in that way, we found that the MPP4s, otherwise known as lymphoid prime myeloid multipotent progenitors, very few are genuinely multipotent. It was less than 5%. And the rest were biased towards output, towards 1 or 2 lineages, B cell only, B plus DC only, myeloid plus DC, myeloid only, B only progenitors that add up to 100%, but they're not all multipotent. All right, so at the end of that study, we published it. It's like, okay, they are different, but the big question is, how are they different and why are they different? And, in fact, this work plus a whole bunch of great single-cell RNA-seq studies have inferred new models of hematopoiesis, where it's not this ball and stick where everything branches, but there's a continuum of states. And in fact, while you draw a box around hematopoietic stem cells, that box contains myeloid-primed, lymphoid-primed stem cells, which follow myeloid-only lymphoid-only trajectories. This is one of my favorite diagrams, and it comes from Martin Guillaume, Milner, and Yona in Immunity. And this is a recent attempt I had in a review, which is pretty much the same thing, just drawn in a different way, where we're used to drawing boxes around populations, but within that box, you get grades of commitment that are part of a trajectory that's already, let's say, myeloid biased from the stem cell, and its daughters go through several phenotypic stages that we know and love, but they're only a subset of each of those stages en route to myeloid development. So this so-called continuous model is something that's really emerging not only in the field of hematopoiesis, but you might have seen published in Cell recently, also in embryogenesis, but to be honest, all aspects of development. It's not going to be strict bifurcation of this. And single-cell RNA-seq is useful to discover things if you can validate it. And this is a paper we published last year in Nature Immunology. So the original experiment, we did 192 cells. We found a cluster and we validated it. And basically, we found a lymphoid-primed progenitor amongst multipotent progenitors. And we validated this, what we saw differential gene expression, but we validated it by using a DAC1 GFP reporter. And you can go read the paper to see how this progenitor only made lymphoid cells, whereas the DAC1 positive MPP4s or LMPPs made myeloid cells as well. So, we found a way to split cells by a marker we would not have a priori looked for. We had to do single-cell RNA-seq and then go validate it. Now, this is the strategy most papers have used. Do a single-cell RNA-seq, find some genes, find an antibody against that marker or a reporter, and then go and discover that subset. But what if you could get both pieces of information in one experiment? So that's the subject of my talk today. So actually, during my PhD with Ken Shortman, we sorted single MPPs in a dish to generate the different dendritic cell subtypes. Type 1 conventional DC, type 2 and plasmacytoids. In a culture system that we developed, bone marrow plus FLT3 ligand gives you the conventional DC subsets. And to our astonishment, the multipotent progenitors, only 50% were multipotent, and the rest, 5% of clones, only made large clones, sometimes 2,000 cells each of just one subtype, even amongst a pool of mixture cells, which could make all subtypes. So the conditions weren't limiting. That cell was primed to only make one cell type. And so what are these molecular programs controlling lineage biased fates in populations much earlier than we would have anticipated to be lineage primed? And it's a sort of Heisenbergian problem. So, the Heisenberg uncertainty principle says you have a particle, you can know its location, but not and its speed, but not both. And it's sort of the same thing with single cell biology, because you can drop a cell in a dish, you can find out what it would make. Let's say in this case, it made orange cells. But then you want to know what was special about the ancestor cell that allowed it to make orange cells. You can't do it because the cell's divided and gone. Alternatively, you can do single-cell RNA-seq, get the gene expression profile, but you've lost the opportunity to now test it for fate because you destroyed the cell. So how can you get both pieces of information? Solutions are either a time machine, where you can test a cell for fate, go back in time, then test it for gene expression, and then link the gene expression and the fate of the same cell together. All right. Don't have a time machine, but what we do have is the ability to let that single cell undergo a couple of divisions so that its daughters become surrogates of the founder. All right. But for that to happen, the daughters need to be highly correlated in their fate to examine that. So long story short, and you can go and read this paper if you don't look at the rest of this talk, we found a way to split clones, test some for fate and some for RNA-seq, or and we call that protocol cis-seq, sisters for parallel fate and sequencing, or cis-skew, where we have normal fate clones, and we can perturb clones to find out what is their fate normally and what is their fate after perturbation. All right, cis-seq. So you drop in a cell, you expand it, then you split those daughters, you test them for fate, or you test them by RNA-seq. And as I said, that's clone splitting for parallel fate RNA-seq. But the DC progenitors need to be conserved for fate. So here are 4 example clones of MPPs. We dropped them in a single well. And as you can see, this clone was biased towards generation of this subtype, which, trust me, is CDC1s. This clone was biased, CDC2s, sorry. This clone was biased towards CDC1s. This clone was biased towards plasmacytoids. And this clone was not biased. It could generate all 3 DC subtypes. So the question is, what did the sisters of this clone that were put in a separate well make? Did it make the same thing or different thing? And the answer is it made the same thing. So those daughters knew ahead of time what they were going to make. All right. So now we know that clonal siblings are not 100%, but highly conserved for their fate. And we tested this statistically by comparing real sisters. Short distance indicates highly correlated fate, and long distance indicates random sisters. So what are the, now we can get the gene programs, right? So we can find the clone that was made plasmacytoids only. We can go back in time to its siblings that we did RNA-seq with at the time of clone splitting and ask what genes correlate with it in terms of fate. And long story short, we found 500 genes that correlate with fate bias towards plasmacytoids, CDC2s or CDC1s. And here they are, all are on one ternary plot. And this is lovely in that it's the correlation of genes with fate, but obviously we want to find causation. So we did a CRISPR screen. It took us a few years, but we finally got it working. And there we take wild-type bone marrow or Cas9 transgenic bone marrow, take the stem cells, we transduce it with a guide RNA library with 4 independent guide RNAs for each of the 500 genes. We culture them and FLT3 ligand, and then we sort for the 3 DC subtypes from wild-type or Cas9 transgenic. And we do a PCR for the guide RNAs and sequence them. Now, if the guide RNA is equally represented, knocking out that gene had no effect. If you knock out a gene and the guide RNAs goes up, it means you knocked out a suppressor. If you knock out a gene and the numbers go down, it means you knocked out a promoter, a gene that promotes development of that cell. And we found we had some spike-in controls, so our internal controls all worked. RUNX2, RF8, TCF4, and Z2 all affected cell fate in the way we expected them, and these numbers indicate number of independent guide RNAs. Here are the ones that we rediscovered with cis-seq. So we knew they existed, obviously, but just through cis-seq, we found these genes correlated with fate, and then when we knocked them out, we found they are causative of fate, but they are. And we found some novel genes as well. And I won't go through all of them today. And some of these we've validated deeper than others. And today I'll talk to you about BCOR, which is this hit here, which is quite an interesting gene. And this is the full list of 30 genes that skewed cell type differentiation one way or another. It's not gene expression here that we're looking at in the heat map. These colors represent the relative increase in proportions of certain DC subtypes over the others. In the interest of time, I'll probably skip this slide, except to say that when we knocked out BCOR, we did see an increase in the number of pre-DC-like cells in cultures. So somehow it was maintaining a pre-DC cell type. These are the progenitors of DCs. And we knew that by surface staining the single cells and doing single cell RNA-seq and finding some unique gene signatures associated with that. And that was all in vitro. So we're like, okay, sorry, I should actually just spell out what the phenotype is. The phenotype is that when you knock out BCOR, you don't have much of a change. You do have a bit of a change in CDC1 numbers, they do go up, but mostly it's CDC2s and PDCs that go up in number. So you're knocking out a suppressor of fate for CDC2 and PDC. And so that was in vitro in our CRISPR screen. And so we thought, okay, let's validate this in vivo. instead of a CRISPR screen, we'll just take single guide RNAs for B core, wild type bone marrow, Cas9 transgenic bone marrow, knock out the gene for B core, transplant them into recipients, do plus or minus FLT3 ligand and harvest bone marrow and spleen and compare the relative ratios of B core knockout versus wild type stem cell contribution to the dendritic cell subtypes. And we've actually first did PBS experiments and we found no difference. We couldn't recapitulate our phenotype. And we tried it once, twice, 3 times, 4 times, same answer. Knocking out B core in vivo did not change the ratio of DC subtypes. I was like, oh, shit. What have we spent our time doing? And then I thought, well, our assay is adding super physiological amounts of FLT3 ligand. Maybe it's that allows B core to reveal its role. So then Sarah injected mice with them without FLT3 ligand in the reconstitution experiments. And then we did skip phenotype. So it turns out that B core suppresses CDC2 and PDC numbers, but only during emergency FLT3 ligand responses. And you see emergency levels of FLT3 ligand in these novel immunotherapies that people are investigating, both preclinically and clinically, by injecting FLT3 ligand. So that's certainly boosting DCs for cancer immunotherapy. You also see FLT3 ligand levels increase with certain viral infections. So it looks like B core is suppressing too many DCs to be produced, and specifically a couple of cell types. Look, I won't go through the data, but it's actually the pre-DCs that I told you about. The pre-DCs are dividing more culture that contribute to high numbers of CDC2s and PDCs. And you can find them in vivo and bone marrow as well. We confirm that with CTV staining that they divide faster and divide more. And I won't go into that data here. You can look at it in the paper. And so at this point, the summary is that B core suppresses PDCs and CDC2s during emergency FLT3 mediated DC development, so that in wild type, B core suppressing output of these cell types. In its absence, these cell types are increasing. All right, but this doesn't tell you at a clonal level, what is the explanation for high numbers of CDC2s and PDCs? Because you can think of, well, you can think of 2 ways to increase cell numbers, but you can think of 4 ways to change cell type fate. So a cell could make more of something, a clone, I should say. A single cell makes a clone, and in a normal setting, it makes a certain amount of cells. And in the perturbed setting, it makes more cells, or it makes less cells per clone. Alternatively, a cell makes cell type A in normal settings, and in a perturbed setting, it makes cell type A plus B, right? So it acquires fate, gains fate potential, or maybe it already makes A and B normally, and when you perturb it, it loses the potential to make B. So basically what I want to say is that clones can contribute to increases or decreases in cell number, either through clonal expansion or contraction, or fate acquisition or fate loss, which is a kind of time machine problem. How do you know what a cell makes ordinarily and what it makes after perturbation? You need a time machine, or can you use SysView? So this is a graphical representation of what I just said. A clone can acquire fate for a new cell type. It could have 2 cell types, have fate for 2 cell types and lose 1 fate. It could expand or it could contract. So how do you find this out for your stem cells of interest? What it makes normally and what it makes after perturbation. So we came up with this idea called SysView. Same principle, molecular time machine, use the sisters in different assays to try and find out what's happening. So I'll walk you through this experiment. Cas9 transgenic progenitors are barcode labeled first, so you can do this at high throughput. Those barcode labeled cells are then allowed to expand in culture for a few days so that you have multiple daughters per clone. Then you transduce those stem cells with B core guide RNA, or a guide RNA of interest, or overexpression construct, or whatever you want. But the point is, if you do it at 50% transduction efficiency, then at random, a given clone's daughters, half will have the gene knocked out, in our case, with cherry expression, and half the clone will not have the gene knocked out, and it will be cherry negative. So now you can sort cherry positive cells and cherry negative cells from the same clone, find out what barcodes they make, and ask, I'll tell you in the slide, if barcode 1 in this case could make all 3 DC subtypes, and when you knock out B core, because they're cherry positive cells here, and you do barcode PCR on the different cell types in cherry, barcode 1, no change. Barcode 2 could not make plasmacytoids ordinarily, but when you knock out B core, can now make plasmacytoids. So it has gained the fate potential for plasmacytoid development. This clone, barcode 3, could make all 3 DC subtypes. When you knock out B core, it loses the fate potential to make one of them. All right. So these different classes of fate change are fate gain, fate loss, expansion, and contraction. I'm just going to look for a time check here. Okay.
You find that you have plenty, at least 20 minutes more than that.
Oh, right. Okay. I'm sort of rushing through it there.
No worries. It's going
Okay.
right. Thank you.
Thank you. All right. So this is the experimental plan. Let's do the experiment. So first we did it on wild type. We took wild type bone marrow, transduced 50%, took wild type bone marrow, barcode labeled them, clonally expand them, transduced them with the B core guide RNA. But because they're wild type bone marrow, no Cas9, there's no gene knockout, it's our control. So we can control for the effect of transduction and cherry expression and the experimental setup. And as you can see, the number of barcodes found in total or in the individual DC subtypes between cherry positive and cherry negative did not change, right? No difference. When you look at the clone size, so a given clone makes X number of cells in the cherry negative fraction. Did the transduction with B core guide RNA in the absence of Cas9 increase or decrease the clones anymore? No. Basically, the dots fell along a relatively straight line. And then you can, that same data you can reflect in a change in clone, relative change in clone size, going down, clonal contraction, or up clonal expansion, and that wasn't much of a difference in wild type. Now, when we use Cas9 transgenic bone marrow, you can see what the absence of B core does to the clones. So interestingly, the absence of B core didn't significantly affect the number of barcodes in the experiment. So it's not like some barcodes were dormant in a wild type setting, and when you knock out the gene for B core, now they become activated. Actually, that's not quite true. They do, and I'll show you why. But the numbers don't reflect that activity. I'll get back to that in a sec. But there was not a gross change in the number of barcodes of cell types. When you look at the clone size, now the cherry positive cells, the B core knockout cells, do relatively expand in size. So all of these clones to the right-hand side produce more cells in the absence of B core than in the presence of B core, which is the wild type cherry negative cells. As you can see the shift, and another way to see that shift is this distribution here, where some clones, quite a few clones, are making 10 cells, a 10-fold greater number of cells for the same clone in the absence of B core. So here we have our time machine. What does a clone make ordinarily, and what can a clone make in the absence of B core? All right, hold that thought. But it's not just clone size, it's fate, right? And fate is multidimensional. It's a little bit of this and a lot of that, or a different ratio of that. So there was no tool out there to actually help us understand what makes B core knockout clones different from wild type clones. So we developed a method called SKU-ID. Tom Weber, developed a method called SKU-ID to analyze Sarah's SKU experiments. And so Tom used multi-dimensional space to compare wild type cherry negative to wild type cherry positive and the reverse, and asked which clones look different between the two conditions. Knockout to wild type and wild type knockout, and sorry, in this case, it's just cherry. So cherry neg to cherry pos and so on and so forth. And there wasn't much of a difference. These red clones indicate something that was different, and those were very few clones with very small output. They really didn't change much, telling you that the system does not perturb fate, which means the system is a fair approach to now test other factors that do change fate and understand how they change fate. So we now do that with wild type and B core knockout. And in that case, we did find quite a few clones that perturb their fate in the absence of B core. And that's indicated by the orange clones here. So clonal skewing with B core, just to remind you, the possibilities of clonal fate skewing. You can gain fate, you can lose fate, you can expand a clone, or you can contract a clone. And when we looked at the data in this heat map, we found examples of a few of those in our B core experiment. So the way you read this graph is every row is a barcode. It was the barcode that we labeled clones at the beginning of the experiment. But the left 3 columns are wild type and the right 3 columns are B core knockout of siblings of the same clone. And the barcodes that we're showing here are detected in CDC1s or CDC2s or PDCs in wild type or knockout settings. And here I've got a little annotation of how B core, the absence of B core, skewed fate. So in these clones, when you knock out B core, all the clones make the same cell types, but they make more of them. So that's why I put this in bold. Multipotent clones make more cells of maximum potency in this assay. Right, so this is an example of clonal expansion in work, in operation. This cluster, on the other hand, you see barcodes that ordinarily, so clone stem cell progenitor cells that ordinarily can make CDC1s and CDC2s, cannot make PDCs. But when you knock out B core, suddenly those clones now release their potential to make plasmacytoids, and you see lots of barcodes present in PDCs in the knockout setting. So B core has somehow released the suppressor of PDC fate in these clones. And we looked at all the examples that we found of how clonal fate was perturbed. And in fact, here's that one example I was telling you about where clones made nothing in wild type settings and then made everything in B core knockout settings. So did the absence of B core release a quiescent signature? We don't know. We don't know. We haven't investigated this further, but I hope you'll agree this is a super interesting way of understanding not just B core knockout, but okay, any gene knockout, but you can overexpress a gene, an oncogene. How does the expression of an oncogene change a fate ordinarily into its cancer clone? Or what about single nucleotide variations? How does a stem cell's SNV determine output? In this case, you knock out that one nucleotide, and how does that output change, with that nucleotide change? You can think of all kinds of examples where skewing fate, cytokines, classic one. What does a cell make in the presence of this cytokine? And if you add another cytokine, what does that clone now produce? Is it really plastic? Does it make a new cell type, or actually doesn't it? And that's a subject for another day. So that from this data, we can attribute the majority of increase in, remember I showed you in bulk right at the beginning of the validation experiments that when you knock out B core, plasmacytoid numbers go up. Why do plasmacytoid numbers go up? This graph tells you that 80% of those numbers are attributed by clones acquiring fate potential for plasmacytoids to contribute to plasmacytoid numbers. But the CDC2 number increase is not contributed by fate gain. And instead, numbers of CDC2s that increase in the absence of B core are explained by clonal expansion. So that same gene, 2 cell types increase in number, but that numerical increase was attributed by gain in fate for some clones, for one cell type, and expansion towards a given cell type for another cell type. Sorry, that's a bit convoluted, but there can be different reasons why cell type numbers increase. It doesn't have to be the same reason. All right, that's the end of the talk. You can look at the graphical abstract in your own time, but just to reiterate the highlights, I think this summarizes it quite well. Clonal multi-omics, that is testing the siblings in different assays, allows you to test RNA-seq in one, fate in the other, attack-seq in one, chip-seq, metabolomics, whatever you want. As long as you can do a single-cell assay, you are now not limited to having to do single-cell multi-omics. You can do clonal multi-omics. And if you've got a clone that makes 10 daughters, you can do a different assay on each of the 10 daughters, link them all together for that one cell. Awesome. CISSEQ, which is our first technique here, identifies the genes that control clonal fate. We do this for stem cells, for dendritic cell fate, but you can imagine finding the genes that correlate with stem cells that produce one lineage versus another, or lots of a cell type versus small numbers of a cell type, or a cancer stem cell, or whatever, right? Anything where clonal heterogeneity is of interest to you, CISSEQ might be an approach to find the genes that control that clonal behavior. CISSQ identifies how perturbation affects clonal fate, as I just described. And we've identified BCOR as a novel regulator of dendritic cell subset development. And I want to acknowledge at this point, this is similar, we've been working in this for quite a while, and we had a preprint in 2018 on this, but Alan Klein's seminal science paper, he doesn't call it CISSEQ, he calls it state-fate relationships. And Arjun Raj has a publication called Rewind, which is using microscopy-based technique, but it's the same principle of a kind of a molecular time machine to find the past correlates of future clonal behavior. And others, Sam Morris is working in this space, Alejo Rodriguez-Francelli, and many others. So I would like to mostly highlight for the talk today, Sarah and Tom's contributions, as well as Yaring, who's not on this slide. They're the three first authors of that paper. Danny, who helped with single-cell RNA-seq. And Lu Yitan, Lu Yitan is the first author. He did all of the bioinformatics in Matt Ritchie's lab that really contributed to this paper. And on the horizon, if I talk in 2 years from now in a forum, I hope to show you what we're really excited about next, which is creating barcodes inside cells, inside a live mouse using a new technology we've developed to demonstrate in vivo relationships. So on that, I will say my thanks. Happy to take questions on Twitter. Don't know when I'll answer them, but happy to take them. And thanks again.
Shalin, thank you so much. This was wonderful insights on the development of dendritic cell, the biology of that, and amazing technological development to dissect the fate of the single cells in heterogeneous population. Thank you so much to explain a very complex topic with such an amazing clarity. You really have amazing communication skills, so we appreciate it. And congratulations on the beautiful baby to you and your wife and your other son. And we will never forget that you have generously given 1 hour of the first day of your son for the Global IMMUNOTALK. So we really appreciate it. So briefly, I will now share my screen here to guide attendees to ask questions via Twitter. So you search for the Global IMMUNOTALK account in Twitter. You will find a tweet that says ask questions for Dr. Shalin Naik here, and then just need to reply to that tweet with your question and mention the hashtag Global Immuno, and Shalin will use his personal handle for answering the questions. And as you will understand, this will be whenever he has the time to do so, because I bet he will be busy in the next few days. So Shalin, thank you so much again, and congratulations for your research and for your family.
Thank you, and thanks to my wife for giving me permission to talk as well today.
Absolutely.
All right, bye-bye.
Take care.
Thanks everybody for joining us, and thanks especially to Shalin and his family in spending this time on such a special day of their life. See you next week. Bye-bye.